Great speaker. I'm beyond than excited to announce Pedro, and I hope that I pronounce your last name correctly, de Magalhães, who leads the Genomics of Aging and Rejuvenation Lab at the University of Birmingham. He also is the scientific officer at the Youth Biotherapeutics and the founder of the Oxford Cryotechnology. Welcome, Pedro.
Thank you very much. So thank you very much, Aubrey, Martin, the organizers, for inviting me here and for this opportunity to tell you a little bit of one of the many hats I wear. So I'm a professor at the University of Birmingham, and I study the biology of aging and different facets of aging, trying to develop interventions for aging. But I wear other hats. I'm also have an affiliation with Oxford. As you mentioned, I'm also CSO of Youth Biotherapeutics. You've heard Yuri Dagin, who's over there, give a talk a couple of days ago. I have a data analysis and consulting company in Oxford as well, called Magellan Science. And I do other also advisory roles, for example, for 199 Biotechnology, whose CEO and CSO are here as well, Boris. So I do various things, and today I'll talk about different topics in this session on low temperature. And I'll focus on cryobiology and cryopreservation, and I'll tell you in the end a little bit about one of the new enterprises I'm involved in called Oxford Cryotechnology. So my goal for today was actually, so what do I want the audience to learn today? And my goal for today is to convince you that cryopreservation is a longevity technology. And so it will, quite a long introduction, most of it is going to be background on the importance of cryopreservation for health, for medicine, and for longevity. So when you think about low temperature, we know that from the field of aging, that if you lower the temperature, for example, of 5 kilotherms, so cold-blooded animals like worms and flies, up to a certain point, they live longer. So there is an inverse relationship between temperature and longevity, which makes sense. And there's even some evidence from mice. So at the lower temperature, chemical reactions, biochemical reactions, they slow down, and so you'll have a slower aging process. Whether it's because of damage or non-damage causes, that's something for a different discussion, but you have a slower aging process. And we also know that some of the longer-lived animals actually live in quite cold environments, like the Arctica islandica, this clam that lives hundreds of years, and the Greenland shark that's been estimated to also live for hundreds of years. And even the amazing naked mole rat, we heard a fantastic talk yesterday on naked mole rats from Andre. So naked mole rats have a lower body temperature as well, which actually we've speculated that it is a contributor to their longevity. So we know low temperature can have benefits, but actually my talk today will focus on even lower temperatures, really freezing temperatures and cryopreservation of biological materials. So I'm a cell biologist by training, and I know that if it's possible to cryopreserve cells, for example. The biggest problem is ice formation. So if you have ice, we can physically damage structures, and because ice expels solids, it can also create patches with high concentrations of solids that create damage. So there's a lot of problems with ice. So to minimize that, we use antifreezes. We use cryoprotectants like ethylene glycol and DMSO in order to cryopreserve biological materials. And there's a number of applications I'm sure you're familiar with. We can cryopreserve cells. As I said, I'm a cell biologist, used to cryopreserve cells all the time. And it's also used in reproductive biology. So you can cryopreserve embryos and then implant them. So the record, last time I checked, this was, I think, broken last year, was of these two twin embryos, two twins that were born from embryos that were frozen 30 years before. So that's quite remarkable. I mean, I'm not sure you could even make the case that that's a life extension. I mean, they were conceived 30 years ago, and then they were alive 30 years longer, and they likely live a normal lifespan. So it's quite remarkable that you can have people that have been frozen, although admittedly when they were very small, and small number of cells. Now, the major focus really then is about preserving large organs and cryopreserving large human organs. That's the ultimate objective. Now, this is not possible yet. We cannot, for reasons I'll mention it a little bit, we cannot cryopreserve large human organs. And that is a huge issue. So I'll give you some numbers from a Nature Biotech paper that was published a few years back. So when you look at organ transplants, there is a huge loss of transplantable organs. So, this would be hearts, lungs, kidneys, livers, but it's particularly serious for hearts and lungs. So, only 30% of transplantable hearts and 20% of transplantable lungs are actually used for transplants. So most of them are actually discarded. One of the issues is that once there's an organ available, there's a very small window of time, sometimes less than 24 hours, in order to find a matching donor. And if you don't find it, then basically the organ goes to waste. So the inability to preserve large organs is a huge problem in terms of organ transplants. So how much of a problem it is, this lack of organs for transplantation, which we heard earlier from Justin, actually? Well, as I'm sure you're aware, there's a huge demand for organs for transplants. And so these are some numbers which actually, when I start looking this in some years back, it's actually bigger than I imagined. So this is data from the US, but every year there's 50,000 additions to the waste lists for organ transplants in the US. But if you calculate how many people die from organ failure, from end-stage organ diseases, it's actually over 700,000 every year in the US. I mean, that's more than, I think it's about 600,000, a little over 600,000 that die of cancer every year in the US. So if we could have organ replacement therapies available for everyone, this could prevent over 30% of all deaths occurring in the US. And it would double the average per person's likelihood of living to the age of 80. So it would be quite a tremendous impact on longevity if we could make organ replacement available for essentially everyone who needs it. It's not just a problem from the US. There are massive needs for organs across the world. I mean, as you can see, so this is lighter colors is larger number of organ transplants per individual, and darker colors is a smaller number. As you can see, in North America, Europe, Australia, there's a way more organ transplants than in other countries, like, for example, in Africa or parts of Asia and South America. So it's a global problem, not just a problem of industrialized countries. So there's a huge unmet need for organ preservation in different facets. It's certainly in organ transplantation. So the point is that if we could safely preserve organs, human organs, we could basically have biobanks. You could basically have organs for transplant frozen or vitrified. I'll come back to the distinction between frozen and vitrified in a second. But you'd have preserved organs in biobanks that you would have plenty of time to match to donors. And you'd be able to save thousands of lives every year. So that's one area. I mean, there's a number of different other applications, including in cancer treatments and fertility, for ovarian cancer, for example, even in cancer treatment. I mean, my wife died of breast cancer who metastasized to the liver. So she essentially died of liver failure. And obviously, someone with cancer, is not a priority for an organ transplant because the cancer would spread to the new liver again. But once you have organ failure in the liver, you can't even do chemotherapy anymore because the chemotherapy is processed in the liver. So, if you could have transplants even for cancer patients, that it wouldn't cure the disease, but it would allow to extend their lifespan. And a number of other applications, for example, in terms of cord blood, in terms of skin banking for trauma victims, regenerative medicine, and even for biomedical research and drug discovery. If we could have human organs, banks of human, not just human organs, human tissues, for drug discovery and for biomedical research, that would be, well, as the previous panel argued, would be better than using animal models. So there would be a lot of advantages for research as well. And there would be a lot of advantages in other fields as well. So, I mean, I mentioned organ banking for transplants. That's one of the major advantages, saving thousands of lives. In reproductive medicine, which I mentioned already, for stem cells, engineer regenerative medicine as well. And when you think further ahead, even for things like more futuristic, like space travel. And human medical biostasis, that is cryopreserving whole human beings that died of a disease or a cause that cannot be cured now, that could be revived in the future once a cure is developed. I think Max Moore will be speaking later today more about medical biostasis and cryonics. So there's a number of potential applications of cryobiology. So what is the current technologies or methods? I mean, this is a little complicated slide. I'll try to guide you through it. But there's essentially, I mean, to simplify things, see from a paper we published a couple of years back, but there's essentially two general ways of cryopreserving biological materials. One of them is slow cooling, or slow freezing, which, as the name implies, you lower the temperature slowly with relatively low cryoprotectants, low concentrations of cryoprotectants. So cryoprotectants act as antifreeze, but of course the problem is that they become toxic, particularly high concentrations. But by cooling slowly at a low concentrations of cryoprotectants, you minimize the amount of ice formed both intracellularly and extracellularly. So that's essential. I mean, there's other things you can use. You can use apoptotic inhibitors. You can use other chemicals to improve the process. But that's essentially, in a nutshell, the approach. The other approach is vitrification. So vitrification is slightly different, basically because it uses a very high concentration of cryoprotectants. So you use a very high concentration of cryoprotectants, which is essentially replaces the water in your sample or in your organ. And so you prevent ice formation by replacing the water by a cryoprotectant. But you have very high concentrations of cryoprotectant that can become toxic as well if there's a long exposure. But the advantage of vitrification is that essentially you have this, it's called a glass-like state that you end up at very low temperatures. So let me show you a figure, actually. This is from one of the pioneers, if not the pioneer in the topic of vitrification, which is Greg Fahey. And this is a vitrified rabbit kidney you see on the right. So you have a frozen kidney on the left, as you can see, with lots of ice. And on the right, you have a vitrified, this is a rabbit kidney, so it's relatively small. It's not a human kidney. It's a rabbit, so it's a relatively small organ. So you have a vitrified kidney on the right, which, as I said, minimizes ice formation. And in these experiments, what they actually did was they vitrified the kidney, then they rewarm it, and then they surgically implanted it back into the rabbit, and they showed it was functional. But keep in mind, that's a rabbit kidney, so it's quite small. So, I mean, if you're interested in sort of what's the current status of the field, I mean, we published a big review with a number of colleagues, particularly Roman Bauer and others, in BMC Biology 2 years ago. So what we know is that we can cryopreserve cells and small tissues, but the problem of larger organs is, well, it starts being a physical problem. It takes longer to cool down and to warm up a larger organ than a smaller one. So there's a physical problem, and because it takes longer to cool down a larger organ, the organ and the tissues and the cells are going to be exposed to cryoprotectants for longer, and thus they're going to become more damaging. So that's I think, a problem of cryobiology for decades now. So we know the problems. We have to prevent ice formation. We can do so to a successful degree with cryoprotectants, but they have toxicity as well. So there's still a number of existing problems in scaling up to larger organs. There has been some advances. I mean, this was on the cover of Science just a couple of weeks ago. So putting life on hold. I think the title was stopping the biological state of an organ in this case. So there has been advances. I mean, this particular advance was actually more on rewarming. So develop a technique for using iron oxide nanoparticles for magnetic rewarming of the organ for rewarming afterwards. And one thing that I find quite fascinating as well, because I love animals and I love unusual animals like the naked mole rat and so on, is that we can also take some inspiration from nature. So there's a number of animals that partially freeze during winter. So probably the best example is the wood frog. So these animals, as you can see, it's quite remarkable. They don't freeze the whole body, but their brain freezes, their heart freezes. It's quite incredible that they partly freeze during winter. So what seems to happen in the case of the wood frog and other animals is that they have natural antifreeze mechanisms, processes that they use in order to resist the damage caused by and to minimize ice formation. In the case of the wood frog, if I'm not mistaken, they use sugars. Well, a lot of these animals use sugars. I think it's just glucose. Basically, their liver, I think it's the liver, basically their liver produces lots of glucose, lots of sugars, and this prevents ice formation. But so just like in the field of aging, where we have amazing animals like the naked mole rats that we heard already, we got to hear as well about bowhead owl and bats. There's also inspiration we can take from nature in the case of, for example, the wood frog. So that's, well, I hope I persuaded you that, at least that cryobiology and cryopreservation is an important topic, an important longevity technology to develop. So what have we been doing along these lines? I mean, I've always found it quite interesting. Now, one of the things, one of the background I come is that I see biology as a data problem. As others have mentioned today already, biology is really, really complicated. Aging is really complicated. So it's about making sense of all the data and all of the information available to try to gather insights and try to progress, which is something we've done quite a lot already in the context of longevity and aging, which I won't go into detail, but we've done quite a lot, for example, in developing data-driven methods, bioinformatics, machine learning, AI-driven methods for identifying longevity drugs. So that's, it's a data-driven approach where we use a lot of existing data, whether gene expression or protein-drug interaction, whatever it is. We take a lot of existing data and we try to predict new longevity drugs. So, I mean, we published a number of papers. We just published a paper I think 2 or 3 weeks ago on applying machine learning to predict new longevity drugs in C. elegans. For example, we published earlier this year on rilmethedin as a new longevity drug that we identified and that we showed it extends lifespan in C. elegans. So our lab, we can say we have a lot of experience in these methods. So what we've been doing is trying to apply these methods, these genomics and data-driven approaches to cryobiology. So one thing we've done in collaboration with Greg Fahey, actually, is we did some gene expression profiling of cryoprotectants. So to me, one of the greatest challenges is understanding cryoprotectant toxicity and how we can minimize it, and how we can develop formulations, chemical formulations that minimize toxicity from cryoprotectants while protecting against ice formation. So we did some gene expression profiling of ethylene glycol. As I said, it's one of the, together with DMSO, one of the 2 most widely used cryoprotectants. We used in one cell line, which was endothelial, vascular endothelial cells. So that's something we've published in cryobiology. But really what we want to do next is we want to do more of these experiments. We want to do more molecular profiles of cryoprotectants, other cryoprotectants in more cell lines, in more concentrations, and then really use some of the methods that we've developed and we've applied already to longevity and to identify new compounds for longevity, to identify, in this case, new cryoprotectants and new toxicity neutralizers, new chemicals that can help develop improved formulations for cryopreservation. So that's what we're working on at the moment, and that's our goal. And so what we've done is we created just a months-old company called Oxford Cryotechnology. So together with Roman Bauer from the University of Sussex and Emil from Tomorrow Biostasis, who's here somewhere. So he's here in the conference as well. So Roman also has expertise in computational modeling and cryopreservation and developing computational methods for making predictions of, in the context of cryobiology and other contexts as well. So we created this company, and our goal is to do this application. So we're starting with cellular validation, in vitro validation. So this is, by the way, that's the Queen Elizabeth Hospital at the University of Birmingham, where I'm based. We're based in Oxford because so Sussex and Birmingham, so Oxford is about halfway between them. And well, because I also have an affiliation with Oxford and already have a company in Oxford, it's quite a good place to start a company. But so we can do the in vitro experiments. I mean, we're at the moment actively fundraising, so our goal is to then do experiments even in animal organs or even in human organs. So that's the idea, is to scale up. So start with cellular approaches, and I'm a cell biologist, so to me that's what makes sense, but then scale up to organs, eventually human organs. So that's the goal. So to summarize, the goal is to improve the cryopreservation protocol. So use some of the computational tools and methods that me and Roman have developed and already applied in a variety of contexts, very successfully, I would argue, to create new protocols that improve the cryopreservation of cells, tissues, and ultimately entire organs. That's our goal. So as I said, I mean, we're still starting. We're looking for partners, and so check out our website and feel free to get in touch. So to summarize, I hope I persuade you that of the importance of cryobiology. I mean, clearly, end-stage organ disease is a major cause of mortality, with thousands and thousands of patients dying every year. Because of limitations in organ preservation, there is still a huge wastage of transplantable organs. As I said, 2/3 of hearts, more than 2/3 of hearts for transplantable hearts are wasted. So that's a huge problem that results in this lack of organs for transplants. So in addition to preserving organs for transplant, there's other multiple biomedical applications of cryobiology in reproductive medicine, regenerative medicine, who knows, space travel and human biostasis as well. But clearly there is a very immediate need for developing technologies for organ preservation and developing organ banking that would allow us to save thousands of lives. And so the problem, of course, is scaling up to larger organs, and so therefore there's this need for developing better organ preservation protocols. One crucial limitation in this context is cryoprotectant toxicity, which is something we're addressing using our genomics and computational data-driven approaches. And so in this context, we created this company, Oxford Cryopreservation, to apply this machine learning AI approaches to cryobiology and ultimately to develop better cryopreservation protocols that, as I said, would have broad applications. So with that, I want to thank you very much for your time and attention. I mean, I'm very easy to find online, but if anyone wants to get in touch, feel free to drop me a line. Thank you very much. Thank you.
5 minutes of Q&A.
Okay, so I think there's a couple of questions here already. They're all in a row, which makes it easier.
Who put their hand up first?
Does no one open the cup?
Yeah. Okay. Thank you, João, for the presentation. I just wonder if you can comment, on the system you use to image biological sample. Because I know, I'm sure you know about this a Nobel Prize in chemistry by the Swiss Dubouchet, who succeeded somehow to vitrify, as I understand, very basic, vitrify water. in the vacuum environment of an electron microscope. So you have a complementary technique to the electron microscopy using this result, which brought Dubuchet the Nobel Prize. So, that's not really the area of cryopreservation, but this is, organ or sample preservation. But I just wonder if which technology you use to study biological samples, so next to the standard electron microscopy, and eventually using this technology, which is a Nobel Prize technology, let's say.
Well, yes, thank you. I think, we use standard microscopes
Right.
where, the photos of cells I showed you. That's an interesting, I'd have to think exactly how we could apply. So we don't use electron microscopes for that. The basic readout we use is cell viability, whether the cells are alive or not. I mean, you can look, because I have an interest in aging, of course, you can do things, and we did in the context of cryoprotectants, we also looked at things like cell senescence, are the cells senescent? And when you stress the cells, you do tend to have some senescence as well. But the primary readout is really cell viability, whether they're alive or not, whether they're functioning or not. That's the primary readout that we employed. Because it's still a problem. I mean, if you freeze cells, a lot of them still die, unfortunately. There's still a window of improvement even at the cell level. And then, as I said, we want to scale it up to larger tissues and organs, even human organs.
Thank you.
Thank you.
Thanks for the great presentation. One question I have is with in vitro testing and cell types. What are the range of cell types you're currently testing, and what are you looking to test in the near future? I guess ultimately you want to move the way up to neurons. So.
Yes, so that's a great question. What I would like to do, every single cell type in the universe, or at least all human cell types. So that are feasible to culture. I would like to do an array of them. We started with vascular endothelial cells. Actually, it was Greg Fay's suggestion, because the vascular system is quite important for organ preservation, right? Because if your vascular system bursts, it's not good. So that was his suggestion. So we started with vascular endothelial cells. As I said, for I think good scientific reasons, but definitely, what we want to do is we want to do other cell types, and ideally different tissues, and that's the ideal scenario. So it's primarily an issue of resources and funding. If we have the resources and the funding to do a vast array of cell types, then absolutely let's go for it. Otherwise, we'd have to prioritize like we've done so far.
Are there any other cell types that you could see as being particularly problematic in addition to neurons for preservation?
Well, I think it depends on what organ you're aiming for. Because I think it's guided by which organs are you going to try to cryopreserve. I would probably, if we go to an organ, it's probably going to be kidney. Just because of practical considerations. There's some data already from Greg and from others using kidneys. You can get rodent kidneys, for example. I could have access to rodent kidneys from the university. So that there's some practical considerations there. So when I think of the way ahead, I would probably keep in mind what organs we're going to do. Neurons, yes, that would be interesting, but so would other cell types as well. Yeah, so yes, a lot of work still to do. Thank you.
Pedro?
Yes.
Question.
Oh, hi, yes. Sorry.
How good would you have to get before you would consider this to be a success? Even things that were very, very good at cryopreserving, like C. elegans and all. There's quite a bit of loss in the rewarming. And my question is, what percentage of cells are going to have to survive? Basically, what would you consider to be a success, even in, let's say, a mouse kidney?
Ooh, that's a good question. I think ultimately you would need to have a functional organ, right? What Greg has done, for example, in his rabbit kidney is I don't remember if he looked at the percentage of cells that were viable. What he did was he transplanted the kidney back into a rabbit and then checked for kidney function in the rabbit. And there was a dip. I mean, it wasn't as good as a non-vitrified kidney. There was a dip. But ultimately, I mean, if the goal is to use organs for transplantations, then you have to go to clinicians and say, you know, what would be the dip in function you'd expect in order to be able to still use an organ for a transplantation. I should say, as a side note, we did a paper a few years back, like a theoretical paper, on loss of organ function during transplantations. So how old can you get a heart, for example, for a transplant? Because obviously organ function declines, and you know, you may not get an organ from a 20-year-old. You know, you may not get an organ for a 40-year-old or a 50-year-old. It depends on the organ. It depends on the health of the individual from which you are getting the organ, the donor organ, right? So there are parameters that are already established for what would be an accepted dip in the quality, in the function of the organ, that you would still be able to use it for transplantation. And I think that is relatively established from a clinical perspective. So that's what I would use. I don't know how to translate it into number of cells. I think that would be interesting, actually, to even maybe do some experiments about. But I don't have a number. I'm sorry.
All right.
Okay. All right.
Unfortunately, we have to. But thank you so much, and
Thank
I really
you.
love the enthusiasm you have for your research. So cool. Very inspiring.